Hello everyone, my name is Rahul. In the previous video, we had discussed about cytoskeleton. In this video, we are going to discuss about cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the substances present around the cell. These substances are usually fluids, flagella, and are responsible for cell movement. Now talking about the ultrastructure of cilia and flagella. Both cilia and flagella they have a similar ultrastructure, which is basically a covering of plasma membrane which encloses a central hub or a central part known as exoneme. So both cilia and flagella they are covered by a plasma membrane. Inside the plasma membrane, a core is present. It is known as exoneme. This exoneme it possesses microtubules which are running parallel to the long axis. This exoneme it usually has nine doublets of peripherally arranged peripherally arranged microtubules and a pair of central microtubules. So we can see in the diagram that this outer covering it is the plasma membrane. On the periphery we can see the presence of these doublets of microtubules, and in the center there is a pair of microtubules. Now this arrangement of microtubules is known as 9 plus 2 array. This arrangement is known as 9 plus 2 array, where 9 represents the peripherally arranged microtubules and 2 stands for the centrally located microtubules. The central tubules are connected by bridges and enclosed inside a central sheath, which is connected to these tubules. So we can see in the diagram that the central microtubules they are being connected by this bridge, and there is this red colored central sheath present around the microtubules. Of course, the color is not red inside the cell. This is only depicted as color red. So this is the central sheath. The peripheral doublets are also interconnected by the presence of linkers. The peripheral microtubules are interconnected to one another by the presence of linkers. These peripheral doublets, they are connected to central microtubules by radial spokes. So in total there will be 9 radial spokes, as we can see in the diagram, that these structures represented by yellow color, these are the linkers or called as inter-doublet bridges, and these represented by black color, they are radial spokes which connect the peripheral microtubules to the central microtubules. Both cilia and flagella emerge from centriole-like structure, basal bodies, or blepharoplast. This is why both the cilia and flagella, they are quite similar in their ultrastructure. Now, talking about the differences between cilia and flagella. Cilia, they are smaller in size, approximately 5 to 10 micrometers, whereas flagella are comparatively larger. They may be up to 150 micrometers in size. Number of cilia per cell is very high. So there will be numerous cilia present in a single cell, whereas these are fewer in number. Flagella are fewer in number. Cilia beat in a coordinated manner. Such a movement is known as pendular movement. Whereas the flagella beat independently. This type of movement is known as undulating movement. Cilia take part in locomotion, attachment, feeding, and sometimes also in sensation, whereas flagella are only involved in locomotion. So with this we finish off with our discussion on cilia and flagella. See you in next video.